Okay, this is the review for Chapter 2, Part 2, when I'm going to focus on organic chemistry and biochemistry. So, basic building block of organic molecules, organic compounds that make up our bodies are carbon-containing molecules. They have a carbon backbone. It's the basic building block of living things. Um, okay, about 18% of your body weight is this carbon, just the carbon. And what's nice about carbon is that it can form four separate covalent bonds. So each carbon molecule can form four bonds. As well, it can form some single or double bonds. So common constituents that come along that bind form bonds with carbon to form organic molecules are hydrogen, nitrogen, oxygen, as well as other, but these are the most important ones. It can form quite a variation of different types of chains. They can be linear chains. They can be branch or even ring shaped. And they can form small molecules. We can call them micromolecules, but more often you find the term macromolecules. So these are rather large compounds, so they can form very, very large um, chains of carbon compounds. Here are some examples on figure 2.12. Here's an example of a carbon dioxide molecule here. And you can see that here carbon is forming four bonds, but two double bonds. So here's a double bond, and here's another double bond to form carbon dioxide. By the way, these are very, very strong bonds. You can form here, for example, lipid molecules or long chains of carbon. And this can continue on, so they can be quite large. And here's a more elaborate or complicated structure of a backbone of an amino acid, which amino acids will be the building blocks of proteins. And so you can see here there's a bond with oxygen, and here's a bond with nitrogen and hydrogen, and then this interesting ring structure at the end. So you can see that the variation can be quite dramatic. By the way, this amino acid shown here is phenylalanine, one of the 20 basic building blocks of proteins. So these macromolecules are synthesized and broken down within cells. They are taken up through the process of digestion, the basic building blocks for making these large proteins or large macromolecules. So basically, to synthesize a lot of large macromolecules, organic macromolecules, involves a lot of the type of reactions known as dehydration reactions. So collectively, these reactions are referred to as dehydration synthesis. And basically, you're removing water from the link of molecules when you're putting these together. This requires energy to build things. And you end up with large macromolecules that originate from smaller subunits. Generally, these reactions, this part of your metabolic reactions, is known as anabolism. And so these are anabolic reactions. When you break down large macromolecules, a very common type of reactions that happen are known as hydrolysis reactions. And for these, you need water. You add water to the chain, the large chain that you're breaking apart. You add water, and you end up with smaller molecules. And this typically releases energy. It's the way you get energy from a lot of large macromolecules. And the branch, the reactions of your metabolic system, your metabolic reactions that are involved in breaking down these compounds are collectively known as catabolic reactions. So we say catabolism. Catabolism is that part of your metabolism that is concerned in breaking down um, large macromolecules. Excuse me. So you can see dehydration and hydrolysis are basically opposite reverse reactions. Figure 2.13 shows this in pictorial form. So here you have a hydrolysis series of reactions this way. So you start with a large macromolecule here, such as this carbohydrate, and as you go this way, you break it down into smaller constituents, and that requires water. So here's water going into those reactions. Here showing you just one of them, showing water going into this reaction, but all of these reactions will require water as you break down things. So here you go from this two component micromolecule here, you end up with two separate individual sugars. This is releasing energy, right? And then the opposite, this way, are your dehydration synthesis reactions, where you're starting out with smaller molecules and you need energy. You have to put in energy into the reaction. You have to put in energy into the reaction, and as you do so, you end up with larger and larger macromolecules. So this is a very summarized version of this. It can be more complicated, but it gives you a good idea of how this works. So we're going to talk about the different types of organic molecules found in your cells. The first type are your carbohydrates, this first class. Carbohydrates are used for many things, but typically they're very important for energy as well as providing structural support to many cellular and tissue components. This is the general formula for a basic carbohydrate. I'm not going to ask you to know this. Within the carbohydrates, the simple sugars are known as monosaccharides because they're composed of just a single saccharide unit. And here's some examples. You've all heard of glucose, very important one in our metabolism, as well as fructose, galactose, ribose, and deoxyribose. These are some of the most important ones to know as examples of monosaccharides. The oligosaccharides are composed of more than one monosaccharide. They're linked together. So if you have two monosaccharides put together via dehydration synthesis, you end up with disaccharides, two monosaccharides linked together. For example, sucrose is when you combine glucose with fructose. You end up with sucrose. If you put two glucose molecules together in a certain way, you end up with something called maltose. And if you add glucose plus galactose, you end up with lactose. So I'm not going to ask you to know this. You should know these as examples of disaccharides. Here's a pictorial representation, figure 2.14, um, of ribose and deoxyribose. And the production of a disaccharide with glucose and fructose to form sucrose, or basically table sugar, by the way. This is table sugar. This is what you add to your coffee. Here you go. This is a dehydration synthesis reaction because we're removing water to form this larger disaccharide. The bonds between sugars are a source of potential energy. And so by putting together sugar molecules into large uh, structures such as disaccharides or even larger ones known as polysaccharides, you're going to be able to store quite a lot of energy for later use. So a polysaccharide can be made up of thousands and thousands of individual monosaccharide units. And these can be joined either linearly or as branch chains typically. Good examples of these are starch. Starches are branch glucose molecules that plants use to store energy. So animals don't have starch. We eat starch and break it down into individual sugars for energy. When animals store energy in the form of sugars, we make something called glycogen. So glycogen is going to be an important reserve of energy for your body. Okay. Cellulose, on the other hand, is another type of large polysaccharide, but this is mostly used for structural support of plants. So the cell walls of plants are composed of cellulose. And this, by the way, is pretty much an indigestible material for us. So when you eat a lot of fiber, you're eating a lot of cellulose, and it's undigestible. So you can't obtain much energy from cellulose. Figure 2.15 represents um, how you make large polysaccharide chains from individual glucose molecules through the dehydration reactions, synthesis reactions that we talked about. Here's a nice micrograph from a cell showing the little starch granules here and here and here and here, all these little starch granules, which are storage depots for energy, right? All these very highly chained, highly branched molecules rich in glucose, and so therefore you're storing a lot of energy in all of these bonds that you're forming. And every time you have a bond here, it's potential energy that's being stored. So a lot of energy being stored here. The next category of organic compounds that we should consider are the lipids. And these are nonpolar compounds and are therefore highly insoluble in water. And there's three major types that you should know are the triglycerides, the phospholipids, and the steroids. Triglycerides are going to be very important, just like a lot of the polysaccharides like glycogen and starch. These are going to be very important energy storage molecules. And we might see later that these store quite a lot of energy. So if the polysaccharides store a lot of energy, these store even more. So they're even more efficient at storing energy. This category of phospholipids here are going to be a very important part of the structural um, makeup of the cell membranes that make up cells, not only the cell membrane on the outside of the cell, but a lot of the organelles that are made up by membranous material inside the cells. Steroids are a very large class of ring carbon structures, and they have a lot of roles. They can form structural um, functions in cell membranes as well, and a lot of signaling compounds are steroids, for example, a lot of hormones. A lot of hormones are steroid based. So let's look into more detail here the triglycerides. They're also known as fats and oils. They're composed of glycerol as well as 3 fatty acid chains. The fatty acids um, can be of 2 types. You can form very saturated uh, triglycerides, fatty acids, and in the saturated condition you have all single bonds between carbons, and this forms something that's very solid at room temperature and therefore form fats. Unsaturated fatty acids, on the other hand, have some kinks in the chain of the carbon, and so this is because of the double bonds that are found there. And this means that these fatty acids are more liquid at room temperature and therefore form oils instead. Fatty acids are stored in large amounts in adipose tissue or fatty tissue. And as you can imagine, these are going to be very important energy storage molecules, just like glycogen. Figure 2.16 shows examples of, here's a triglyceride here, and you have a glycerol group attached to 3 fatty acid chains here. Here's one, here's another one, here's another one. And as you can see here, these are saturated fatty acids. They're very linear. They're all single bonds. On the other hand, if you have a triglyceride that is composed of fatty acids that have double bonds, such as here, 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 etc., this forms kinks in the chains, and you can't pack these as closely together as you can these ones. So these tend to stay in more liquid state. Phospholipids are slightly different. You have glycerol, same glycerol, but instead of 3 fatty acids, you have 2, and the third part that's attached to the glycerol is a phosphate group that can vary, that has different types. The phosphate group is relatively water soluble, so it's a part of the molecule that's hydrophilic, whereas the other 2 fatty acid chains, as we've learned, are hydrophobic. These are going to be very, very important components of cell membranes. Here's an example of this, figure 2.17. Here's a phospholipid with the 2 nonpolar fatty acid chains. Here's our glycerol part, and here's a phosphate group compound that forms this polar head. And because this is hydrophilic, it can interact very nicely with water, whereas this part does not interact with water. Okay, and these are going to form the lipid bilayer that form the membranes of your cell. The other type of lipids that are important to know are the steroids, composed mostly of 4 carbon rings. And there's many steroid-derived compounds that we should be aware of. Cholesterol is a steroid-derived compound, as well as the hormones estrogen and testosterone, as well as others. And we'll talk about these when we visit some organ systems as we progress through the course. There's other types of steroids that we'll talk about. Here's a picture, figure 2.18, showing you a steroid-derived 4-carbon, 4-ring carbon structure derived from a steroid. This happens to be cholesterol. I'm not gonna ask you to know the specific formula or molecular structure for all of these. But you can see here from cholesterol you can synthesize various other hormones and compounds such as estrogen and testosterone. The next major type of organic compound are the proteins. Okay, and these are quite varied. Of all the biomolecules, are probably the most varied type. They're very complex structures, and they're made up of amino acids, individual groups of amino acids put together. They form long chains called polymers. And of the amino acids, there's 20 types. There's 20 types that form proteins. There's a few different other amino acid types that don't form proteins, so they're not structurally important. They play other roles, but these are the main ones we talk about here, the 20 amino acids. Most of them you can synthesize yourself. Some of them you have to obtain from your diet. Amino acids have an amino end and a carboxyl end, and various R groups that will give the molecule or the particular amino acid its name. So each of the 20 different types has a different type of R group. When you put together amino acids by what are called peptide bonds, which are a type of dehydration synthesis reaction, you're going to end up with large chains of amino acids, and if it's large enough, then we call that a protein. Here is a table 2.19. You're not going to need to memorize this table, but okay. If you can see here, this is alanine, isoleucine, leucine, methionine, phenylalanine, etc., etc. They all have different names. And if you can see here, the basic structure is the same in all of them. There's a carboxyl end here, and an amino end right here, and a carbon. And then here is the R group. And the R group is what changes among all of these amino acids. And each of these R groups is going to give different chemical characteristics to all of these amino acids. And depending on the order in which you put these together in a chain, in a protein, you're going to have a protein that has various functional capacities. So, very neat. Okay, so in figure 2.20, you see 2 amino acids, isoleucine, and here's an example if you're making a peptide bond here. You have isoleucine and alanine already bound together by a peptide bond. And here you go, here you have another dehydration reaction. You're going to add a valine. So now you end up with isoleucine and alanine, alanine and valine here, forming this polypeptide. So as we mentioned, proteins are complex structures. They're put together by peptide bonds of all those amino acids. And if it's a small chain, we call it a polypeptide, just as like the one we just saw. 3 to 100 amino acids can be considered a polypeptide. But anything larger than that tends to be called a protein. Proteins have various functions, and the function that a particular protein plays depends on its structure. And we talk about the primary structure and secondary and tertiary and quaternary structure of proteins. The primary structure is very simple. It's basically the sequence of amino acids that form a particular chain. And these are, again, all made by peptide bonds. But that particular chain of amino acids can form various conformations that are known as secondary structures. And there's two major types. There's an alpha helix structure and a beta pleated sheet. So both of these are important secondary structures that are formed. And there are some examples of this following. The tertiary structure is more of the three-dimensional shape that is made by all of the secondary structures that come about from the primary structure. So the primary structure defines the secondary structure. The secondary structure, how those alpha helices or beta pleated sheets come together, form a larger three-dimensional, more complex thing, and we call that the tertiary structure. Bonds that are important in stabilizing these tertiary structures are disulfide bonds, as well as the hydrogen bonds, which we already talked about when we talked about the inorganic chemistry of water, etc. The quaternary structure is when you have two separate polypeptide or more polypeptide chains that are associated with each other. So you might have, one polypeptide chain here forming a tertiary structure. So this three-dimensional thing here that I drew is a tertiary structure of one polypeptide chain. But you might have maybe another polypeptide chain here in blue. Might look a little different. So these two can interact in various ways. Here are little bonds showing these two units interacting with one another. And so in this case, this large protein is composed of two peptide chains, 1 and 2. And so how they are put together is the quaternary structure. Now, not all proteins are made up of subunits like this, so not all proteins will have complicated quaternary structures. Here's a pictorial representation of what we just talked about, figure 2.21. Here's the primary structure. It's just the sequence of amino acids with the peptide bonds here. And the secondary structures that they can form, they can be alpha helices or beta sheets. It can also be a random coil, just like a spaghetti. And how all of these motifs are put together in the large three-dimensional structure of the protein, then we end up with what's called the tertiary structure. So here you see an alpha helix, and then a random coil, then another random coil, and then forming a beta sheet, etc., etc. So this shape of the protein is known as the tertiary structure. Okay. And if you have several subunits here and here and here of a very complex, large protein, then we talk about the quaternary structure of this thing here. Okay. Okay, so obviously the function of a protein depends on its structure and its both primary, secondary, tertiary, and quaternary structure. When you heat up or subject proteins to extreme pHs and various other conditions, you can disrupt the structure of the protein. And when that happens, we say that we've denatured the protein. So the process of denaturation breaks up that very orderly structure that we just talked about. So temperature and changes in pH are very important in causing denaturation of proteins. And basically this is what cooking does to proteins. An important class of proteins in your body are known as enzymes. And enzymes are proteins that are involved in bringing about biochemical reactions. And they are basically acting as catalysts. So they speed up the rate of reactions and themselves are not part of the reaction. They're not altered or consumed or modified in any way. They're just there to speed up the chemical reactions. They interact chemically in certain ways, in various ways, but that's what they do. Most biochemical reactions in your body and your metabolic reactions are brought about by enzymes. And if we didn't have these enzymes, these reactions would proceed way too slow, and life would not be able to happen. So a lot of these enzymes are going to be important for a lot of homeostatic mechanisms and just to support biological function. So enzymes are very important. Next, we're going to talk about nucleic acids. Nucleic acids are basically involved in metabolism of genetic information, so storage of genetic information and processing that information in various ways. Nucleic acids are long chains of subunits called nucleotides. So they're not amino acids, they're not fatty acids, they're not sugars. They're more complicated structures, but the individual units are known as nucleotides. There's two types of nucleic acids. There's the DNAs, the deoxyribonucleic acids, and the RNAs, or the ribonucleic acids. DNA is important because it contains instructions for producing RNA. Okay, and then the RNA contains the instructions for producing proteins that we just talked about. And as we just mentioned, the proteins are central to most life processes. So this is important to understand how proteins come about, right? The DNA specifies the RNA, and the RNA specifies the proteins. A nice analogy to this is a cookbook, right? You have a your favorite cookbook, that's all your recipes. I'll try to draw a book here. Okay, your nice cookbook that you love, your favorite recipes are on here, and you want to make a dish, a yummy, delicious dish, your favorite pasta, for example. Here, okay, it's making me hungry. All right, so you don't want to take this nice book to the kitchen because it can get spilled, it can get dirty. You don't want to be using it. So what you do is you you make a copy of a particular recipe, and then you end up with a little working copy of your recipe, right? And you don't care what happens to this. You know, you got your instructions, and you work in the kitchen with this, and you make your dish. And once you're done, you can throw this away. Um, but you know, your nice book remains intact and unharmed. So that's a nice analogy of this. The cookbook being the DNA, your working recipe here is your RNA, and your protein is your final dish. The nucleotides that make up these nucleic acids contain a 5-carbon sugar. So they use sugars to make nucleotides. So it's a more complex biomolecule. In the case of the DNA, the name of the sugar is a deoxyribose. And in the case of RNA, it's a ribose. In addition, each nucleotide contains a nitrogenous base as well as a phosphate group. So they're more complicated molecules. And here are examples of this. Here are the four nucleotides that make up DNA. Here's adenine, thymine, cytosine, and guanine, and they're abbreviated as A, T, C, and G. So here you see a deoxyribose 5-carbon sugar attached to a phosphate group, and then this region here. And so you've noticed that this region is different in all of them. And so this is what specifies what kind of nucleotide you're going to end up with. To make a DNA molecule, a deoxyribonucleic acid, by the way, it's double-stranded, so you can have 2 different chains that come together. And they contain the deoxyribose, the nitrogenous bases that we just looked at. And these are going to pair with one another in this fashion. Adenine always pairs with thymine, and guanine always pairs with cytosine. So there's a picture next to show this well. Okay, here we go. So here's this double-stranded DNA molecule. It's helical. It's forming these helices, and there's 2 strands of them. And here you go. Here's the sugar part of the nucleotide, right? The deoxyribose, and the phosphate groups are helping attach each adjacent nucleotide to one another. And here's the defining part of the different nucleotides. And as you can see, the A here is always paired with the T, and the C is always paired with the G. The structure of RNA is a little different. First of all, it's not double-stranded in most cases. So there's some examples of double-stranded RNA that have unique functions, but we're not going to worry about that now. Most of the RNA that we're going to be talking about is the single-stranded RNA. And instead of deoxyribose, the sugar is ribose. And the nitrogenous bases are pretty much the same as DNA with one exception. So you have the adenine, the guanine is the same, the cytosine is the same. But instead of thymine, you have uracil. So uracil replaces the T in the DNA molecule. Okay, so it's slightly different. And here you see an example in figure 2.25 of an RNA, single-stranded RNA molecule. Last but not least, another important biomolecule found in your body is ATP, also known as adenosine triphosphate. So you should know that. It's a very important compound. It's basically the way energy is converted and moved about in the cell. So it's the equivalent of energy, is your currency. Of energy inside the cell. ATP, the adenosine part, is a nucleotide, as you probably have identified. And the phosphate groups between, the bonds between the phosphate groups in the adenosine triphosphate, so they're triphosphate, there's three phosphates there. The bonds between those phosphate groups is where the potential energy is contained. So when you break down those bonds, you liberate or release quite a bit of energy. So here it is, ATP, rather unstable molecule, and there's three phosphates here. If you lose one phosphate, you end up with adenosine diphosphate. You went from triphosphate to diphosphate, from 3 to 2, plus this 1. And that liberates energy. Here's a pictorial representation of that, figure 2.26. Here's your ribose and your adenosine, right, the nucleotide part. But here's that triphosphate chain, part of the ATP, right? And then as you see here, there's the three phosphates, and through a hydrolysis reaction, you lose one of these phosphates, and this releases energy to do useful work. And in order to store energy, you go backwards, right? You do a dehydration synthesis series of reactions and go from ADP to ATP. Right. Okay, and then in later chapters and later sections, we'll talk about how you get ATP and basically respiration, cellular respiration. Is going to be an important way to make ATP that will then be available to fuel reactions, give enough energy. Okay. Okay, I think that's the end of this review of this part of chapter 2. If you have any questions, let me know.